Hey guys, it's me. It's time for a video on mutations. Just fair warning, the animals are being loud, so they may bark, and I may have to yell at them. I apologize. In your journal, you can write mutations homework notes and put today's date, whatever that may be. And this is just a quick review. In class, we have talked about the central dogma, so just wanted to review it really quick. This is how you should have written it in your journal. So DNA, which makes up our genes or holds our genes and makes us, who we are, it replicates, which is why it's in a circle, means to make a copy of the DNA. So it replicates, and then the DNA is turned into RNA through transcription, and then RNA is turned into proteins through translation. So we have gone over those three processes in class. This is being slow. Hold on. Right, so we've gone over these. We're good. So remember that when you go from DNA to DNA, if you're just making a copy of DNA, you're using the bases A, T, C, and G. So A and T pair together makes the word at, C and G pair together. But in transcription, when you go from DNA to RNA, all the bases in RNA, instead of being a T, they're now going to be a U. So thymines turn to uracil. So the matches in RNA will be A and U, C and G. So the T's become U's. And then in translation, which is the last process, mRNA is turned into tRNA, or they match, using A, U, C, and G. So again, we're in RNA, so we have U's. And then tRNA matches with amino acids using the triplet code. So you'll take 3 bases at a time and you'll match it with an amino acid, string all the amino acids together, make a protein. Alright, so that was just a little review. You're welcome to write this down. If for some reason you don't have it, you definitely need to know this stuff. So if so, you can pause the video. Alright, now the point of this video is really in reference to mutations, which we are going to talk about in class, of course, but simply a mutation is when there's a mistake. So it's basically a mistake in the DNA. It's when it's changed. So a base somewhere in the DNA is changed, and therefore it could, or it does, lead to a change in the mRNA. It could lead to a change in a protein, and therefore it could lead to a change in a trait. As you will see, if one of these letters gets messed up, that means it's going to change one of these letters, and sometimes it changes the protein, and sometimes it changes the trait. Not always. You're going to see that in a second. But you should write this down. This is starred. You do not have to draw this picture. Okay, so there are different types of mutations. There's something called a point mutation, which is when there's a change in just one letter. So it's at just one point in the DNA. Again, it may change the protein, and here's some examples. So if you look at the 3 letters, the 3 bases, right, they're called codons. These aren't real codons, but we're just gonna write them in English so it's a little bit easier to understand. So this says, the fat cat and the red rat ran. But if you change a T to an R, this will now say, the fat car and the red rat ran. Or the A from the original DNA could be changed accidentally to an E, and now it says, the fat cat and the red rat ran. So they're not super messed up, but they obviously don't make a lot of sense. So this would be a point mutation. It's a 1 base change. So if in your journal you just want to write CAT, arrow, CAR, that'll represent 1 base changing. So there are types of point mutations. So you should have point mutations written with some facts, and now underneath that there are types of point mutations. One is called a nonsense mutation, which is when the change leads to a stop codon. So here's the DNA, UAC, which would have coded for tyrosine. Now it's been changed to UAA, so the C changed to an A, but this now codes for a stop. So that means that the protein is going to stop really short. It's known as a nonsense mutation, and it basically means the protein ends up short. So you need to write this down. Another type of point mutation, so stay under that, is a missense mutation, which is when it changes the amino acid. So CGA becomes CUA, and instead of alanine, we now have valine. So that's going to change the protein, right? If you swap out an amino acid, it's going to change the protein, and maybe you'll have blonde hair instead of brown hair. Who knows? So you're welcome to write all this. The important part is that a missense mutation means the amino acid is changed. Here's a real-life example of a missense mutation. For your red blood cells, if you have a mutation in your 6th amino acid in the hemoglobin protein and it becomes valine, then you have what's called sickle cell anemia, which is when your blood cells look like this. It's really hard to carry oxygen, it's hard to exercise, you can become really sick. All right, almost done with point mutations. There's one more called a silent mutation, which is when there's a change in the base, but there actually is no change in the amino acid. So CGA becomes GCA becomes GCU, but they both code for alanine, so it's called silent because there's no change. Nothing really happens. The question is, if there is a change though, why doesn't the protein switch? And your answer has to do with what's called a wobble, which we'll talk about in class. But basically, it's because there are a lot of different combinations that code for a certain amino acid. So even if you change the last base, it'll still code for the same thing. All right, so you've done point mutation. That's the big category with some examples underneath. And now there's one more big category, which is called a frame shift mutation. And that's when you add or delete a base. So instead of swapping a base, one is either taken away or added in. It's also, well, it's called a frame shift because the DNA shifts one direction or another, changes all the codons. Basically, it results in big changes. So if we look at the fat cat and the red rat ran, now if we insert an N between the A and the T, put an N in here, now this T is in this codon, and this D is in this codon, and on and on and on. And now it says, the fat can, tan, the, era, dra, tra, n, which makes absolutely no sense. So hopefully you can recognize that a frame shift mutation is really bad and will result in huge changes in a protein and could be very bad for the organism. So again, under now this category, there are some types of frame shift. One's called an insertion or an addition, which is when you add a base in, which is the one we just looked at. Changes the protein a lot, obviously. And then there's a deletion, which is when you lose a base. So a base disappears. So if we look up here, if we, let's see, if we get rid of this C, the C disappears, the A moves over, now the U moves over, everything moves over. It messes up all these amino acids, and now the protein's really different. So how much does it change the protein? A lot. They're both really bad. Here's an example of a real one. If you have a deletion in your CFTR gene, it can lead to cystic fibrosis, where you don't have control of your body, which is really bad, obviously. All right, and so now in class, we will practice all these mutations. So make sure you have your journal ready to go. Good job.